everyone, and welcome to the webinar today on pangenome annotations and resources in Ensembl. Yes, I'm the Ensembl data flow coordinator. And what that kind of means is that I'm involved in all of the data that is absorbed by Ensembl, generated by our pipelines, and released through our websites. So it does mean that I can be a bit of a jack of all trades and an expert of none. I'm happy to try and answer all questions today, and anything that I can't answer, I will take away to my colleagues. You'll see today that I'm covering a broad range of topics and a lot of work done by a lot of people. So I would be happy to provide a kind of Q&A feedback resource that could be accessed through the website following this webinar. So over the next 30 to 40 minutes, I hope to walk you through very briefly what pangenomes are, because this has been covered in the rest of the webinar series, but we should touch on it again, why they matter, and then how we're representing, annotating, and integrating them within Ensembl. So as we say, this session is part of the broader pangenome webinar series, and today I'm going to focus on how these concepts are implemented in large-scale data resources, in particular Ensembl. So here's what we'll cover today. So we'll start with the motivation, why pangenomes matter, the scientific and practical reasons. We've moved beyond single reference genomes. And as I say, I know you've covered this probably already, but let's touch on it again. And then we'll look at the pangenome landscape and Ensembl's role in that. So the different projects, assemblies, and annotations, and other resources that we currently host. After that, I'm going to go into some detail on how Ensembl annotates pangenomes. So this is where I'll focus on some examples, mostly human, mice, crops, and livestock, and the strategies that we use to transfer and validate annotations across multiple assemblies. And then finally, we'll finish with just a brief thought on what's coming in the future, so the challenges and opportunities, and in particular, the future goals for Ensembl. Let's start with the fundamental question. Why do we even need pangenomes? And firstly, what is a pangenome? You likely know at this stage, but let's just reiterate that a pangenome captures the collective genomic content of multiple individuals within a species, or it could be within a genus or within, whatever level of set of genomes you want to look at. So instead of a single linear reference, we represent variation, and this can be both small scale and structural. And as a connected graph of sequences. So the core parts of the genome then are shared among all individuals, while accessory or variable regions represent unique genes or structural rearrangements. And as if you've been following the series, you'll know this concept kind of first emerged in bacterial genomes, as well developed there. But now with long-read sequencing and improved assembly methods, it's really revolutionizing our understanding of vertebrates and plants. So in a pangenome graph, every node represents a sequence segment. Every edge defines how those segments connect across genomes. And projects like the Human Pangenome Reference Consortium are using these graph structures to provide more accurate inclusive reference. So this reduces bias and allows more precise variant calling. Within Ensembl, then, we're developing the infrastructure to host, annotate, and visualize these multi-assembly resources, and that's what I'd like to talk about. But why do we need the pangenomes? So I've just kind of focused in on three major motivations for building pangenomes, and these cover human health, agriculture, and biodiversity conservation, because this is what we are most kind of connected to within Ensembl. So for human health, traditional references like GRCH38 emit population-specific variation and even some functional genes. So by moving to the pangenome, we reduce that bias, we improve clinical variant interpretation. And as mentioned in the 2023 Nature paper from the Human Pangenome Reference Consortium, they demonstrated that this by building a reference from 47 diverse individuals, which represented 94 haplotypes. And they discovered that there was thousands of structural variants and novel exons that are actually absent from GRCH38, a very trusted, manually curated gene set. So those differences directly impact variant calling pipelines, gene annotation, and ultimately our understanding of disease mechanisms. Then in agriculture, pangenomes capture the genetic diversity among breeds and cultivars, for example, identifying alleles responsible for yield, disease resistance, or environmental resilience. And by integrating these data into Ensembl, then researchers can explore diversity within species like cattle, pig, wheat, or rice, for instance. And then, of course, finally for biodiversity, particularly important in conservation, pangenomics underpins population-level studies that allows us to assess adaptive variation, inbreeding, and evolutionary potential. And this is essential for managing species under environmental change. Okay, so just to highlight three application domains that are, really bring pangenome concept to life. Obviously, precision medicine and pharmacogenomics, where a patient's genome can be interpreted within the context of the full spectrum of the human haplotypes. So a variant lies when a variant lies in a region missing from the reference but present in the pangenome, it can now be annotated and interpreted accurately. This means more reliable clinical pipelines and better discovery of rare disease genes. Sustainable food systems then is where pangenomes enable comparative analyses across breeds and cultivars, as we mentioned. And then we can identify variants that underlie drought tolerance, feed efficiency, or resistance to disease. And this is critical for climate resilient agriculture. In the example workflow here, you can see that data flow from breeding programs through genome sequencing and assembly and into pangenome comparison and trait discovery is all very beneficial. Apologies, I'm just getting over a cold, so I'm going to be drinking now and then. So finally, in biodiversity genomics of conservation, sequencing individuals from endangered populations and comparing them through pangenome graphs can help us to detect loss variation or adaptive alleles, and this can inform conservation strategies. Okay, so let's move on to how are they represented, how these pangenomes are represented in Ensembl, and what resources we already provide or what we're kind of working on developing. So Ensembl now hosts thousands of genomes, and I'll go into a bit more detail about where these are hosted in a second. But yeah, that's thousands of genomes overall, and hundreds of these genomes together form pangenomic resources for key model and applied species. So we actually work in collaboration and support many of the ongoing pangenome projects that you may or may not be familiar with, things like the Human Pangenome Reference Consortia, the Mouse Genomes Project, an upcoming Rat Pangenome Project, the Anopheles Reference Genome Project, and in the agricultural space, you have the Panoriza for rice pangenomes, working closely with Grameen, which hosts crop plant pangenome sites, like dedicated pangenome sites. And then, of course, Eurofang, which are covering all of the farmed animals. And this is to name but a few. There's so much going on in this space. So we've annotated a lot of things. As I say, we're hosting a lot of genomes now. For instance, we have more than 460 human haplotype assemblies have been annotated, 17 mouse strains, 20 breeds each for sheep and pig, with 14 Medaka, and there's some salmon and stickleback as well. 54 Drosophila and 36 Anopheles. Then for crops, we have 77 barley cultivars, 19 wheat, 16 rice, and there's old cultivars on the way now. So you can see we're representing this wide range of pangenomes, and this is growing in breadth and depth continuously. Each of these projects involves multiple assemblies or subspecies genomes, and this is enabling that intraspecies comparative analysis. So all of these projects feed into our annotation, comparative, and visualization programs, and this is what makes Ensembl a pretty good place for exploring genome diversity. Okay, so I'd like to highlight at this point, and I think it's really important to try to give people a heads up on this, that Ensembl is actually currently in a transitionary phase. So we have created a new site, beta.ensembl.org, and we're currently migrating everything to that new site. But because we are in this transitionary phase, it means that depending on what data you want to find right now, you might need to visit our new site, as I say, beta.ensembl.org, or one of our domain sites. So these will be the ones that, if you're familiar with Ensembl, you're more familiar with. So ensembl.org, or say plants.ensembl.org, etc. So depending on what you're studying, you may want to visit beta.ensembl.org, which is our rapidly evolving platform for new assemblies and early access to data sets. So it's updated every 2 weeks with features and the newest data to come off of our pipelines. So if it's something new you've never seen before on Ensembl, you want to go to our new website. So we're working to migrate everything from the older Ensembl sites to here, and we're also adding all the functionality that you know and love from Ensembl over the years, but also so much more. And you'll understand that this is a huge task, so please bear with us while we're working on this. And in the meantime, if you want to go back to something that's stable, it is a production site and has those established comparative tools, gene trees, and multiple alignments for that legacy set of species, this is capped. But there is a legacy set of species there and has all of this functionality, then you can go to ensembl.org. Of course, this is updated quarterly, but these updates will stop after next summer so that we can focus on our new site. Similarly, domain sites like plants.ensembl.org, metazoa, fungi, etc. These are the domain-specific portals that are gradually being unified into the beta platform, but at the moment you can still go there to find those established comparative functionalities for a select set of genomes that are within these sites. So yeah, so as I say, currently you can browse the new site. There you can access data files via the FTP, and I'll go into a little bit more detail about that now. While all of the domain sites share the same core infrastructure so that you can browse, download, and query data using APIs consistently. Okay, so just to go into a little more depth about these access routes, where can you go and get data for your genomes that make up these pangenomes? So beta.ensemble.org, you can view linear assembly. So I should point out the browsers, this is how browsers work currently. Coordinate systems are viewed in a linear way. We're not yet got the functionality to view graphs. But this does mean that you can go to your individual haplotypes or strains that make up a pangenome and explore them using our genome variant pages. And as we move towards that graph visualization, these are the same data sets that will underpin those interactive views. So if you go to the new website, you will see this species selector page on the homepage, and there's lots of icons for what we call the popular species. So these are obviously the things that are most visited, and they have a number on them indicating how many genomes are available for each of these species. So you can see here, it says there's 31 genomes available for mouse strains, and this covers 17 different strains. And if you can't see your species of interest, I should just point out, sorry, in the popular 42, you can just go to the search bar and enter a query term, and this will give you back the results from our over 4,000 genomes that are now hosted in the new website. So yeah, clicking on an icon of choice or entering your search term will return a list of matching genomes, of which you can select the one you want to browse. So here I've selected CHM13, the TT2 assembly from the human genomes, and I can browse it here in a linear view. And we are working on building those comparative views of genomes side by side, including gene neighborhoods and structural variants. So there's like a lot of exciting things to come. So watch this space. In terms of getting the data then, so this is especially for people working at scale, I guess. We have the Ensembl website that remains, the Ensembl FTP site, sorry, that remains the main entry point for downloading annotations, assemblies, and metadata. So you'll find everything organized here by species. There's a README file to kind of describe the directory structure and data fields. And we also expose this thing called species.json. So this is to help you programmatically receive the data that you want. So you will find in there information, so metadata related to, say, the scientific name, the assembly accession, etc. And so you can access that file to tell you exactly where the files of interest are. Yes, and then all of the same data that comes from that FTP obviously feeds directly into the Pangenome projects, and this ensures consistency across all of the data sets. So let's get a little bit more into the comparative genomic resources, because these are the ones that really bring pangenomes into context, and these are accessible through ensembl.org and the domain sites. So let's start with gene trees. So in Ensembl, gene trees represent the evolutionary relationships among gene families across assemblies and species. They're built using one representative protein per gene, aligned across genomes, and then reconciled with the species tree. Nodes are annotated then as either speciation events, so obviously divergence between species, and these are shown in blue, or duplication events, which are shown in red. Gene trees allow you to identify orthologs and paralogs, explore gene family evolution, and crucially for pangenomes, ensure consistent annotation across multiple assemblies. So when we annotate several haplotypes or strains, we can link each gene model back to a single evolutionary node, maintaining coherent gene IDs and orthology relationships. And this framework is really powerful for pangenome projects because it distinguishes between true novel genes and simple duplicates or copy number variants. I know that this is particularly popular. The gene trees are particularly popular in the plants research world. Okay, so alongside gene trees, Ensembl provides multiple genome alignments, so pre-computed alignments across species or assemblies that reveal conserved and variable regions. We use several methods depending on the data set, and this is just a legacy thing. You know, over time, new methods have been explored and implemented. So you will find that for certain data sets, you'll have different methods applied to generating these alignments. So Makata Pecan has been used for global alignments guided by syntety. We have EPO, which can handle rearrangements rearrangements and duplications, and also reconstruct ancestral sequences. And then something that a lot of work was put into, because it requires a lot of resources and it was quite a difficult one to produce or to implement, was the progressive cactus, which is graph-based and scales to hundreds of genomes. So this is really ideal for large pangenome sets. And this is implemented on the Ensembl infrastructure. And we've collaborated with a lot of groups in terms of a lot of communities who've asked us to generate these alignments. And we can do this. We have the resources and we can do this at scale. So it's something that we've been collaborating a lot on with people. Of course, these alignments form the foundation for conservation scores, constrained elements, and ancestral reconstructions. And in the pangenome context, they allow direct comparison between assemblies, so we can see where structural variation or insertion and deletion events occur. And this is all kind of like leading into our development of new views like structural variant views and gene neighborhood views, etc. Yeah, so as I say, these can be viewed in the browser, or for things like the cactus alignments, you might want to access HAL files for downstream analyses. These can be found in the FTP, but as I say, if you can't find what you're looking for, this is something we collaborate with people on. So do contact the help desk because we can kind of consider a community's group of genomes of interest, identify those with you, and then generate these alignments, produce the files and put those on the FTP that can be used for lots of important downstream research and analyses. So it's always good to kind of keep in touch with Ensembl and let us know what it is you're interested in and what you're working on. Okay, so that's where you can browse or download the data. So that's kind of like Ensembl's resources, where you can get them and what you can get. But how do we actually create the annotations across so many assemblies? And perhaps that's what people are interested in today. So I'm going to dive a little into the pipelines that power Ensembl's pangenome work. I'm going to run through some of the examples. And as I kind of highlighted at the beginning, this is like a really broad set of things that are happening across a lot of teams. So I want to just highlight kind of the kind of most important things that are going on and give you some examples of those. So for crop plants, we import community annotations. In humans, we're mapping from this reference. In livestock, we're using our traditional pipelines and lines of evidence. And then I just want to make a final note on the mouse pangenome because for this we used a combined approach. Okay. So for crops, Ensembl's approach here is really community driven. So apologies. We work closely with crops genomics consortia like Grameen, the Wheat Initiative, the rice and barley communities, and we identify species and assemblies of interest. We import their community provided annotations then into the Ensembl infrastructure. They're validated, and then we integrate them into our comparative genomics pipelines. And this collaboration ensures that Ensembl doesn't reinvent existing high quality annotation. Of course, this is much more sustainable. And we do rely on a lot of the expert communities to provide Ensembl with the best data possible. So instead here, we harmonize and extend annotation data. So a key component that I just want to highlight as well here is that this pan-taxonomic or plant compara analyses. So this is kind of specific to plants. And this resource builds comparative gene trees and orthology relationships across broad taxonomic ranges, not just within species. So it includes representative genomes from all major clades and plant Ensembl plants, and including the crop plants. And by using the same homology framework as Ensembl Core, PanCompara connects gene families across hundreds of plant species. So this is like a really high view of the relationships here. It's invaluable for pangenomes, obviously, because it shows whether genes appear novel in one cultivar, whether genes that appear novel in one cultivar are truly new, or they're just unannotated homologs of known genes elsewhere. It supports detection of core and accessory gene sets, highlights lineage specific expansions, and identifies candidate genes for domestication or adaptation. So when you open crop pangenome in Ensembl plants, you're not just seeing isolated genomes, you're seeing them embedded within this pan-taxonomic context. Okay, and just to also note some work that was done within Ensembl and in collaboration with Panorizer is the Magic 16 project. So the Magic 16 panel is a platinum standard set of 16 high quality genome assemblies from diverse Asian rice cultivars. So this captures breadth of variation across, I believe, aromatic groups. Using these assemblies, the Panorizer team then developed a dedicated pipeline, Get Pan Genes, to define what we call pan genes. So that's gene clusters that span cultivar-specific variations and shared loci. So just kind of really briefly, for each gene model across those cultivars, they identify synthetic orthologous regions, grouped them, and then assign stable pan-gene identifiers so that downstream users can trace like this gene cluster across all 16 of the cultivars. And I guess how does Ensembl tie into this? Well, the Ensembl developed this method that you can read about in the paper mentioned on the slide here, and then annotated the assemblies and pan-gene identifiers which have been imported into Ensembl plants and the Beta Ensembl website. So within Ensembl, each cultivar's gene models are cross-linked via this pan-gene cluster, and this enables uniform comparative queries across the cultivars. It means actually that we can integrate multiple cultivar strain assemblies under unified cluster identifiers, improving comparability. It supports downstream analyses like trait mapping, variation screening, presence and absence variation, and of course gene family expansions. And it improves annotation consistency. So if a gene appears in one cultivar but not another, the pan gene ID helps flag that absence as biological rather than an annotation artifact. This has been a really key part of pangenome work in the plant space. Okay, so then for humans, our work aligns with the Human Pangenome Reference Consortium, as I mentioned, and all of this work has been described in the Nature paper in 2023, where they produced reference quality assemblies for 47 of individuals representing 94 haplotypes, and these were from really variable populations around the world. In release 2, then the HPRC covers 232 individuals or 464 assemblies, and so there's a massive representation going on here, and this is just growing and growing. And Ensembl, then, we have annotated all of these haplotypes, and we do this using a projection-based approach that transfers high-quality annotation from GenCode reference onto each of the haploid assemblies. So we take this carefully curated subset of the GenCode gene and transcript set, and that's carefully curated in that we exclude things like read-through genes, so which span 2 adjacent loci, and any genes that are only present on, say, patches and alternate haplotypes. And the reason for this is because they're complex and ambiguous loci, which would confound mapping. And so instead, we focus on this well-supported, stable subset of the annotation. For each gene, then, we build what we call an anchor sequence. So this includes the gene itself and a defined amount of its surrounding genomic context. And the flanking sequence provide this kind of unique positional information that helps us locate the equivalent region in a target haplotype assembly. So this is a bit like, I guess, taking a street address plus its neighboring landmarks to find the right location on a city map, because obviously there are multiple streets with the same name, so that those neighboring landmarks provide that context. Okay, so once the likely corresponding region is identified, this is where we perform pairwise alignments from the reference region to that region that you've just identified on the target. And that alignment is used to project exon coordinates and intron structure, and then of course your other gene features onto the new assembly. So this gives us a direct base level mapping of the GenCode model onto each haplotype, but of course we've kind of bypassed doing very expensive whole genome alignments here to use the synteny. After that primary mapping, we perform an additional genome-wide search to catch recent duplications or collapsed paralogs that might not have been detected in the one-to-one alignment. So here we take each canonical transcript from GenCode, align it across the target genome. And if it finds a new mapping that doesn't overlap an existing annotation, that's a clue that the gene may have a duplicate copy in that particular haplotype. So something that's extremely relevant in pangenome context, where gene copy number can really vary between individuals. So when we evaluate the results, we typically see 3 different scenarios coming out here. So the scenario 1 is the ideal case, obviously, and of course, well, luckily, actually, this is by far the most common, and this is where we get perfect mapping from the linear reference to the haplotype. So the intron exon structure and the transcript boundaries all align cleanly, and the annotation could be transferred directly within onto the haplotype with minimal adjustment. This is what we expect from the majority of genes that are stable and well conserved across the human haplotypes. But of course, there are other scenarios. And in scenario 2, this is where the alignment software begins to struggle. So this can happen in regions that have large structural variability, such as copy number variations, segmental duplications, or these long repetitive regions. So simply where this, or simply where the sequencing quality in a particular haplotype might be lower, although with these kind of last consortia, assembly quality is just improving. So this is not so much of the problem anymore. Okay, so in these cases where we're seeing the software begin to struggle, the anchor sequence might find these like roughly corresponding regions, but the local alignment then can't reconcile those differences. And those annotations at the moment is where we flag things, and we hand these to our manual annotators. And I'll touch on that a little more in a minute, but we're also working on incorporating individual specific transcriptomic data to correct things. But yeah, this is all in development, so there's still things we need to do. Finally, then there's scenario 3, and this is where we have partial or missing mapping. And this is where parts of the reference gene just simply don't exist in the haplotype. This can be because that sequence is absent or because it's been structurally reorganized to the point that the alignment just isn't meaningful. And this can reveal true biological absence of certain genes or exons, or it can point to assembly gaps or unresolved repeats. So in the human pangenome, these unmapped regions are actually some of the most interesting because they often highlight those structural polymorphisms and population-specific insertions or deletions that were invisible in GRCH38. So overall, this mapping pipeline lets us systematically project a trusted reference annotation onto each haplotype while still detecting structural divergence. It ensures that we can annotate dozens of human assemblies consistency, which is obviously really key within the pangenome. And this links each gene across haplotypes for a shared gene code identifier. So I mentioned the kind of get pan genes way in the plants of naming these clusters at the moment through human. This is all in development, but for now we rely on those gen code identifiers from the parent as it's mapped to the haplotype. And we also record where variation creates new or missing features. So this is the foundation of Ensemble's human pangenome annotation. It's a hybrid of projection for stability and alignment-based discovery for diversity. And as I mentioned, there are things along the way. I mean, this is automated mapping, and it can introduce artifacts. So some loci, particularly those with segmental duplication or high copy number variation, are really challenging to map correctly. And as I said, for these, we just flag them and pass them to the GenCode team, and they do this targeted manual annotation. So at the moment, they're focusing on CHM13 assembly, where we have mapped annotation to that, and they're creating, so they're focusing in on these places that we know are kind of difficult to annotate or where they're finding incorrect mappings, and they're creating through manual annotation what we're calling blessed models. So basically, these blessed models would serve as gold standards if we came to do re-annotation or remapping because we have a new gen code set, or improved data to do something here. We know not to touch this because this is taking manual annotation. So by combining automated projection with expert curation, we're retaining this high coverage while ensuring biological accuracy. And we really are developing this kind of feedback loop and figuring out, how this can most efficiently continue to annotate the human pangenome. Okay, just to touch on the livestock species then, because we just kind of treat these as we would any kind of separate genome, in that we run the full Ensembl annotation pipeline for vertebrates on this. So this combines transcriptomic and protein alignments to produce the best evidence model at each locus. And these pipelines generate gene sets from multiple breeds or subspecies. And what we're trying to do here is produce consistent gene models across the assemblies. Of course, that's not like all that easy. We do kind of try to use the same set of transcriptomic data for a species, and obviously always the same protein data from the databases to align to the genomes, but there's variability in terms of the assemblies and things like that. So we are as consistent as possible in terms of the gene sets. And then we can apply our comparative tools. So as I noted before, we generate gene trees and multiple alignments within each species, and this links homologous genes and highlights diversity. It supports breeding research and trait discovery in things like cattle, pigs, and other domestic animals, and keeps our friends at Eurofang very happy, gives them a lot of data to use in their downstream analyses. So yeah, just as I said, a final note for the mouse pangenome consortium, who released their first full comparative analyses this year. So they assembled genomes from multiple inbred strains. I think I mentioned there's 17 of those. And they showed that nearly 10% of mouse genes exhibit structural or copy number variation between strains. So in Ensembl, our job here was to annotate these, and we started by using our human mapping pipeline. And we mapped from GRCM39 reference and found obviously that we were missing some things. And this was a lot of work that was done by Jintel, who was at Sanger at the time. And he came back to us to show us how much novelty is expected between these strains these strains that obviously we didn't expect based on what we'd done with the humans, but these strains are more diverse than human haplotypes. And it really cemented the fact that we needed a combinatorial approach here. And so we integrated strain-specific transcriptomic data to identify the novel isoforms and strain-unique genes. So this is like our dual approach here. We've got mapping and novelty capture, and this ensures that we don't miss new functional transcripts that only appear in certain strains. Obviously, this data is crucial for interpreting genotype-phenotype links in mouse models, and all of the annotations kind of contributed to that publication there and the ongoing work for the mouse pangenome. Okay, so that's the what, the why, and the how, I guess. So let's have a look at what we're doing to look ahead. So what challenges remain and what opportunities lie ahead for pangenomics in Ensembl? So pangenomics sounds exciting, I guess, but it's really not easy. The challenges are both technical and conceptual. On the challenges side, I guess representing graph-based pangenomes efficiently is still hard. Most tools assume linear coordinates, as we've seen with all of the browsers still. There's a lack of agreed standards for formats and metadata as well. I mean, this is an issue across genomic annotation in general, but particularly in the pangenome sphere, I guess, and graph-based and work. Tooling is still immature, I guess, particularly for visualization. We do have these really nice pieces of software now that are generating these lovely graphs. How do we integrate that into the Ensembl infrastructure has been quite a challenge. And as we add more assemblies, we need scalable ways to manage annotation consistency. So I mentioned like that we would have blessed gene models from the manual annotators, and we wouldn't touch them again. But what about if we use the graph to kind of bless whole regions within the graph? If we know this is identical across all of these haplotypes, so we don't have to come back and add more evidence to find the variation here. We're pretty content with what's been done to reduce the resources to kind of do reannotation. And where do we kind of stop in terms of what is a release of a pangenome? Do we add more and more haplotypes on top of this, or do we kind of stop at a point where we say this is our pangenome and the representative therefore? But of course, the opportunities are huge. In human health, pangenomes will improve clinical outcomes by reducing reference bias and uncovering hidden variants. For agriculture, they'll inform breeding, and for food security and climate adaptation. Biodiversity, then they'll enhance our understanding of how genomes function and vary across the tree of life, obviously help us with these conservation efforts too. And for research infrastructure, they'll push us towards interoperable graph genome standards, which is all great opportunities. The goals for Ensembl then, well, these align closely with those opportunities as they should. We are expanding our pangenome coverage first and foremost. As I've indicated with our new website, we have over 4,000 genomes, so more species, better tooling, and improved integration. These are the key things. We're working a lot on interoperability, so we've connected with initiatives like GAF and GBiz and other genome graph projects. So GAF, the Genome Annotation Framework, defines schemas for describing annotations on pangenome graphs, enabling exchange between databases. GBiz then is the Genome Browser Interoperability Zone, which standardizes APIs across major browsers like Ensembl, UCSC, and NCBI. And both of these aim to make graph-based genome data interoperable, so annotations in Ensembl could be visualized or queried in other tools without this translation issue. And we're also tackling visualization head on. So we're developing intuitive ways to explore multi-assembly data. I kind of touched on, we've been working on a structural variant viewer. And we're also considering kind of like gene neighborhoods, how we would view those. So all of these are very exciting, but very difficult to implement because, as we've mentioned, about that kind of linear coordinate system is what we're very much used to, and kind of comparing those and moving into a multi-coordinate system is something that blows our mind a little. We're also integrating pangenomes within our variation and regulatory pipelines, enabling this population level annotation and truly comparative pangenomic genomics. And there's always kind of presentations and training coming out of the Ensembl group on the variation and regulation pipeline. So if you are interested in those, keep an eye out. I guess at this point I should thank Ensembl, GenCode, and the EMBL-EBI teams, and then all of our collaborators across HPRC, FANG, Grameen, and the broader genomics community, all of which, like, without whom, you know, none of this would be possible. There's a lot of data, there's a very broad set of analyses and work that's going on here. So thank you to everyone. And then I guess thank you for listening. I'm very happy to take some questions. And as I say, I'm happy to provide resources following the webinar as well.
Thank you so much, Leanne. Sorry, I was soon coming out of a cold. So we already have a few questions that came in the Q&A, so we can start with those. And of course, if you have other questions, please just keep on typing and we'll try to take as much as we can. We do have 20 minutes, so I think we can cover quite a few questions. So the first one, more than a question, is actually a clarification, since it came in really in the last minutes of the talk. I believe at some point you used the phrasing novelty capture because someone is asking, what do you mean by that?
Oh, so for the mouse pangenome, so perhaps I should have touched on that a little more of the diagram here. So yeah, so as I say, we started out by annotating the mouse pangenome in exactly the same way as we had done the humans. So we took a mouse strain and we just projected things from the GenCode mouse gene set onto that assembly. And of course, you can't find anything through projection that doesn't exist in the GenCode set. So as Jintao kind of started to dig in with things he knew from ab initio gene finders and the transcriptomic data that he was aligning, he started to see these novel things that were coming up. So there's novel genes here, and this can be from copy number variation. And sometimes he was digging down into a gene and seeing new exon, intra-exon on structures. So basically, yeah, he was seeing within these strains here these new things that just weren't seen within the reference. I believe the reference is this one in the middle here. So without going outside of the reference and doing something different, we were never going to find these. And that's where we brought in that strain-specific transcriptomic data. So the transcriptomic data was taken specifically from each of the strains. When we mapped that on, it obviously kind of found these novel regions, these new regions. And when we combined everything together, we had a much more complete gene set per strain then. Does that make sense?
Yeah, thank you. And I would actually take another question that I believe is connected somehow, because now you say without going out of the reference genome, we would have not find this. And someone is asking, what would you say are the advantages of looking at pangenomes instead of just looking at reference genomes? So is there something else you would like to add on this?
Yeah, well, it's that key point that we are stuck with reference bias, right? So take GRCH38 or GRCM39. It comes from this one sample. Now there are a few individuals that contributed to that sample, but they're very few. It's not representative of all of the populations or all of the strains, right? And there is variation from one human population to another. There's even wider population from variation from one mouse strain to another, and therefore the reference which we work from should be representative of all of that. And as you start to add more and more individuals to this reference, which is now becoming a pangenome reference, you see more and more of these novel things come in, these variations come into there. So it's kind of combining all of the things that you find from all the different places into one reference, right? So I hope that answers the question. Yeah, there's bias in the reference because it comes from one source. But we've always relied on that reference. We've done a lot of manual curation on that reference. It's very high quality. So it's a good starting point to map onto these newer strains, haplotypes, etc. But it won't find the novelty. We need to look to that strain specific, that individual specific transcriptomic data that comes directly from them to find these new things, these variations.
Great, thank you. We can move to a more technical question. So this is related to the getPangenes. So one of the attendees is asking, does getPangenes cluster by synteny also? How are structural variants handled in those cases?
So sorry, the first part, does it cluster by synteny? Yes, I can answer that. What was the second part, sorry?
The second part is, how are structural variants handled in those cases?
I'm going to have to go away and ask someone and say, as I said, this is all very broad, and I don't work specifically on plants, and this work was done by someone who has moved on from EBI, so I haven't even had the chance to collaborate with them. So it's a really useful tool that I think people should look into, and I wanted to highlight. And there's the manuscripts there, and people can go and have a look at it. But in particular for that question, I will go away and ask my colleagues in the plants team to put together a much better answer than I can instead of me waffling on about something.
Perfect, thank you. We can then share this in the webinar page so the people can find it. Thank you so much. So we can now move to the next question. So someone is asking how and what resources are available to delve into pangenome constructions and annotation in Ensembl. Oh yeah, this is just given that this is the last webinar in the series. So yeah, I think.
Yeah, so pangenome annotation, as I say, from an ensemble point of view, we're doing this in a linear way. So all of our pipelines, which are available, they are all open source. They're quite difficult to do outside of ensemble infrastructure, although we have provided some support in the past. And I would like to highlight as well, over the next 2 years, we have received funding to make all of our pipelines deployable. So that's all great news. So people should be able to use ensemble tools to do annotation. This will be on the linear level. The next step then is that downstream thing of doing the comparative analyses, right? So that's how you can bring together things for these pangenome analysis. And we do provide some of the comparative analyses for you. As I said, we can do cactus alignments, which is quite difficult for a lot of people because it takes up a lot of resources. So you contact us if you've got ideas about that. There's HAL files available through the FTP. So if you want anything that we've annotated, it's obviously available for our website, for our FTP. Things that we've used in our comparative analyses are all available, which is helpful for your downstream pangenome studies. And then I guess there's lots of tools out there for constructing these pangenome graphs, which is maybe what people want to look at. So PGGB, oh, my friends in the comparative would kill me for not knowing the names of all of them. But again, this is something I can provide in resources after. It's like, here's all these links to things that you can start to build your own pangenome graphs from. So all of these are very useful. So I'd say in terms of what Ensembl offers, you can get annotations from us, you can get multiple genome alignments from us, and then you can apply downstream analyses to these. And I'd be happy to provide a list of tools that helps you with that. So I hope that answers the question, because I don't think you, like, just to kind of point out, you can't go to Ensembl and download a pangenome. But what would that be? Would that be the graph? Would that be the multiple alignment? So I guess you have to think about, like, what is it do you want? Do you want the annotations of a select set of subspecies and then the alignment for that? Do you want to build a graph from that? So yeah, there's some downstream analyses to do, but we're happy to support that as well, and do contact the help desk if you need help with it.
Thank you. The next one is quite general, so I guess it's also more a perspective on your side. So how do Ensembl pangenome annotations support comparative genomics and evolutionary studies?
Yeah, so sorry, just repeat it for me. So how do Ensembl pangenome annotations support other people's comparative
Compar
studies? Yeah.
yeah,
So
comparative genomics and evolutionary studies.
yeah, so in general, as I say, we provide over 4,000 annotations on over 4,000 genomes. So I guess it's supporting there is that you could come along, you can take our genomes, the gene sets that are there, and you can do those downstream comparative analyses. It's all open source, all freely available. In particular, in terms of consortia, we work together with these huge biodiversity consortia, things like the Vertebrate Genomes Project, where they've got these 581 phase one genomes that they've assembled to the highest quality. We're annotating them in the most consistent way that we can. And then they go into these like cactus alignments or downstream phylogeny building tools. And so this is all facilitating those downstream comparative analysis. So there are targeted kind of consortia that we work with that we're kind of contributing to doing doing the analyses in terms of running comparative genomics. But there's also like the fact that you could just come and take our data. Like all of the data there in open source, so you can do your downstream analysis. So I hope that it's kind of fueling that in terms of which is generating as much data as possible. And also, as I harp on about a bit, but do get in touch with us if you have a community that has a group of genomes of interest that would benefit from us running a calculus alignment. We have the resources. Let's do it. Let's generate those. And if it benefits the downstream community, give us a shout. As you see from, say, the mouse pangenome, we did all of the annotations. They did all of the comparative analyses themselves. But in terms of, say, there's a large Lepidoptera project, so that's butterflies and moths. We annotated over 200 Lepidopteran genomes, and we did a huge cactus alignment, provided the HAL files, and that all fed into a Lepidopteran pangenome study there. So that's, I guess, how we're fueling it, providing the data for it, just providing data at scale. We have the resources to generate this data, so hit us up if you need it.
Thank you. Looks like a huge effort indeed from all the different species you are working with.
Yes, it's really broad.
Yeah. Yeah, someone was asking about bacterial pangenomes.
Mhm.
So if by using, for instance, I guess since you're from Ensembl also, would there be some resources on that as well?
Yeah, so this is not my area of expertise. I'm eukaryotic annotation, but we obviously have Bacterial Ensemble website, which will all be migrated into the new infrastructure. So everything's going to be merged together. And we also work very closely with the Magnify team. So this is the oh God, um Mags. Oh God, I can't remember. Anyway, the Magnify team who work on all of these beautiful metagenomics and all the bacterial genomes, and there's lots of pangenome stuff going on in that space. But I can't speak to it. I don't know about the methods that are used there. But we have the data. Again, we have lots of annotations that are imported from community for the prokaryotic world, but also this consistent this pipeline that's been generated on the Magnify side to choose references from these large amounts of bacterial genomes that they have, and then annotate these in a consistent manner. So again, that's kind of contributing to having these consistent gene sets that can be compared across a pangenome. So I'm not sure that that answers the question. Again, I will go back and get you all the resources for that would better help with it, because it's not my area of expertise. But there is resources within Ensembl if that's any help.
Thank you. Just to clarify, if someone doesn't know the name, so Magnify is another resource, data resource at EMBL-EBI that is focused on metagenomics data.
Genomics, thank you.
Ah, that was the word you were looking for. Okay, sorry. I
Max,
thought it was a different
M-A-G.
one. I don't know why, I thought you were looking for a specific name on one organism or something. And it's like, okay, I think I cannot help, but yeah, I could have helped, sorry. Okay, so the next question. So someone is asking if there is any sequencing method that best work if then we want to have like pangenome analysis downstream.
Yeah, so I guess again, assemblies is kind of ahead of what I do. I like the Sanger, the teams at Sanger are like best people to ask about this for really detailed information, and I have contacts so I can get information on this for this feedback. But yeah, since the kind of invention of long read sequencing, it's really kind of pushed these things forward, right? But assembly quality in general has gone up, and it's through these combined approaches, these deployable pipelines, I should say, that Sanger have created as part of the Tree of Life program, where they're drawing on Hi-C data and all of the other technologies that are coming through in assembly at the moment, and it's this combined approach. And then of course there is that they have this kind of curation level on these assemblies. So it's just these higher quality assemblies that coming through all of these consortia and these programs. I would just say that the best place to look is to those Sanger deployable pipelines and what would you say, the standards and the guidelines that they're setting for all of these consortia, particularly things like Darwin Tree of Life, Vertebrate Genomes Project, all of these, they've set standards now, and they're really awesome. I wouldn't say that assembly is fully solved problem, but yeah, there are these galaxy pipelines that people can pick up and use. I think that doesn't really help if the person is asking here, what technologies should I spend my very valuable funding on? But perhaps though it's like having a look at those Sanger pipelines would kind of give you an indicator of what you should be focusing on there.
Yeah, thank you so much. Sometimes just knowing where to look for the information, it's it's the most important. So thank you. Okay, so there is one question, so I'll take it because it's been also voted. So someone is asking, what are the advantages of using a graph pangenome instead of a representative sequence pangenome? So I would be happy if you like to take this, if you have some comments, but I also like to specify that the webinars we had last week on the 5th of November was specific about pangenome graphs. So maybe this person can also be interested in going back and watch the recording if he's interested in pangenome graph, but happy for you to comment as well.
Yeah, it's just kind of from my point of view, what I was saying about you could have these areas on the pangenome graph that you know are kind of at the collapsed, you know, everything is the same in this region, is very well conserved and everything. And it sort of gives you this indicator of there's not much else what I need to do here. So this is from a very annotation point of view. Whereas in the parts of the graph where you've got a lot of variation and there's this indicator of a lot of structural variation, you would need to use sort of different methods to target how you do the annotation there. As I was saying, it's not going to be a perfect alignment of just kind of lift this over to here because everything looks the same. It will be, I need to consider that, you know, there's some things have been flipped, some things are missing. And we're still actually, we're struggling with those regions. And that's where the manual annotators are really coming in to help us at the moment. But yeah, I think that's the kind of key with the graph is that you would lower resources instead of having to work through everything over and over again. You highlight this is done, this was easy, or you know, here's a region that's quite complicated. You're going to have to use a different method there. So yeah, it's good at informing what we should do in terms of annotation. And then I guess from the point of view of downstream analysis, yeah, it really highlights that kind of presence, absence, variation, like all of the things in one nice visual representation, right? Instead of kind of having to scroll. I guess if you've got kind of like genome A to genome B to genome C, and you have like those syntenic views, your structural variation views, it's quite difficult to see the full
Picture.
picture there.
Mm.
Yeah, it's quite difficult to see the full picture. It's just one, it's kind of almost pairwise. Whereas within the graph, it'd be really cool, imagine if you could kind of look into a graph in a 3D kind of way and look all around the different parts that make up this ball of variation. And then the collapsed regions, as I say, are nice, easy ones that we bless and say, this is done, we are happy here.
Thank you. So the next one is, what are the best practices of polishing the pangenomes and their annotation so that they can be more reliable than the reference genome? And say,
Mhm.
I'm in a view of genome-wide studies applications. So
Okay,
there's
the best practices for polishing. Oh, I wouldn't call any of this polished so far. So I should, yeah, something I always say in the presentations I give as well is annotation is not a solved problem. It's all over the place. And as you see, there's so much variability. So I say the best practices so far and what we're doing and we're still kind of learning from is that feedback loop. And it's manual curation is still so important. Like manual annotators, please don't take away their funding and take them all away from us because yes, we have all these computational resources and we can do a lot with automation now. We can do it at this really large scale without that human input from those really key experts. You know, there's still so many things. Even finding the problems is still a really big challenge. So I think in terms of like, yes, the how did you put it? The kind of like the way to polish things is to still have that manual intervention and feedback loop for it. Because obviously, the more feedback you have, the more we can kind of work on edge cases. And as I say, that to decision path that like here's something I know about this region. This is how I should work on annotating it. And there may be things that like I don't know, we can't do in an automated way. I don't, there might not be the tooling yet, and someone has to really look at this. So manual annotators, I think, are really key in terms of polishing.
Thank you. The next question, I believe is actually related because someone is asking, are the differences in annotation quality or completeness between the reference genome and the alternative haplotypes in the pangenome?
Yeah, so this is a difficult one because we kind of measure quality based off the reference, right? We see that as the gold standard. But what we're seeing, so particularly with CHM13, so this is the T2T human assembly that's come out, and this is the one that the manual annotators are really digging into. That obviously there's gaps in the reference GRCH38 that aren't in T2T. So if you base the kind of quality and completeness of T2T on the reference, it's artificial. Because it actually has more than the reference. So yeah, so it's quite a difficult one to answer. I would say for the most part, you know, so we do it at the moment in terms of how much were we able to map from the reference to this haplotype. You know, 99.9% of the things are being mapped successfully. So we say like, yeah, these are really high quality. This has got everything. And I think kind of filling in those final little gaps and answering the final questions will be with, as I say, the manual annotators and the haplotype specific transcriptomic data. And then And then what do you do? Do you move to the new reference and base all of the quality on that? Um, so yeah, quality assessment can be different because we do it from such a reference-based point of view. So it's never going to be better than the reference because we consider that the gold standard. So I guess it will evolve the more we learn and the more data we put into it.
Thank you. Uh, then we have, if you have any other comments on standards or best practices for working with pangenome annotations in Ensembl.
Standards and best practices. It's all still evolving, so it's difficult for me to say you must do it this way. I think, um, as I say, we're trying to create all of these deployable pipelines. So I would hope that that sort of gives us some kind of consistency at least. Um, but yeah, it's such a difficult one to answer in the world of annotation. Open source data is good. Looking to this kind of manually curated gold standard kind of level of things. Maybe I could have a think about that and come back with like what's good guidelines for best practices here. Um, so yeah, it's a difficult one for me to say. I don't like to be the authority on that as well.
You can simply provide your, perspective and what you think. Doesn't have to be the revealed truth, as we say. Yeah, the next one is also quite broad. So once again, you can just provide your perspective and ideas, and it's connected to a similar one that we had before. So someone is asking, how will pangenome analysis revolutionize phylogenomics, and what potential challenges are there? So potential challenges, I believe we also discussed them a little bit at the end, but there is something that you like to add.
Yeah, I guess this kind of would be kind of key in the pan taxonomic area that I spoke about here. So like what's going on in the Comparative team in terms of building these pan taxonomic phylogenies, and it is really kind of grouping these pangenomes together, but then exploring what happens across the space rather than just within. And as you say, this can kind of highlight is something missing or just unannotated. We find it elsewhere and it just happened to be unannotated here. So it starts to highlight things there for us. Again, I don't want to waffle on too much, so I'll get a better answer from the Compare team on that one. I'm not a phylogenists, but yeah, sorry about that.
Yeah, no problem. Thank you so much. We have a last question and then a few comments that I will take after. So the last one is it's actually connected, I think, to another one we had before. How can I annotate a pangenome graph for a bacterium that is not included in the Ensembl release? Is there any workflow that I can run locally?
Annotated pangenogram. The only thing I know about annotation in the bacteriosphere is Proca, but it does sound like this would probably relate to what Magnify are developing or have developed in terms of their very standardized pipelines for doing these kind of things. And as I say, they kind of take a cluster of things, reduce this down to a reference. So I guess you can consider that like the pangenome representative, and then you could map that onto other things. But I think in the bacteriosphere, I thought things were like genomes were quite small and simple and figured out with Procker, but I don't know too much about that tool itself. I believe within the series as well, the webinar series, there was someone speaking about bacterial pangenomics, right? So it's always worth looking
Yes.
back on that. But again, I would point you to Magnify and I can get my colleagues to answer more questions on that. I know very little about bacteria.
You had the eukaryotic session, so you're entitled to know less about the bacteria. Someone commented just on this question, say you can use PGAP too. The
Okay.
tool is like use PGAP, so apparently it's another tool. So I want just to mention it live. And yeah, so we've taken all the questions. So thank you so much. Leanne, I would like to finish with some comments that we received. So someone is saying, thank you so much, Leanne. It was a wonderful talk and your range vocabulary is quite impressive. So no problem for missing the metagenomic side. And then again, awesome presentation. So I would like to thank you again. And I would also like to thank all the people that have joined for this webinar, for the whole series. So we've done this pangenomic series for around 1.5 months, and the audience was really, really engaged. We had lots of questions, a lot of people attending. So really, thank you so much, everybody.